Hi, this is Amit Vasanji, Chief Technology Officer at ImageIQ. Today I'd like to talk about ways that you might be able to improve your high-content cell protein screening. Traditionally, to assess whether your population of cells on a cell culture surface area is positive for a particular protein or stain, a technologist will review a slide on a microscope, randomly taking images across the culture surface area or looking through the oculars and then counting with a cell counter. This can create a number of errors. It can be imprecise, it can cause, it has issues with accuracy, and then it can underrepresent your sample depending on the number of images or fields that you analyze. At ImageIQ, we take the approach of using a method called large field of view imaging, which essentially raster scans across, using a traditional microscope, raster scans across an entire cell culture surface area and takes high magnification image tiles and then stitches them together. This creates a very large image, but it's high enough resolution for automated image analysis. And then, so what you see here on the screen is an example of that. It's an image of a cell culture surface area that's comprised of 100 image tiles taken at about a micron per pixel. The cells are labeled in, all the cells are labeled in blue with a nuclear marker called DAPI, and then the protein of interest here is red. And this is just a high zoomed up image of a particular portion of that previous large field of view image, and you can see that the protein of interest is actually sometimes located directly over the nucleus or surrounding the nucleus in the cytoplasm. So our approach to doing this analysis in an automated fashion is to segment out the nuclei and then dilate them, which is essentially growing them so that you're now entering into the cytoplasm where some of the corresponding red protein is located. And we do this in a way that doesn't combine or join together the cells, so you can still count individual cells. Once you have that mask, you can apply it to your red channel and then determine the number of cells that are positive for your marker. So this is a image that's generated automatically. It's a pseudo-colored representation of the cells that are positive shown in red and the cells that are negative shown in yellow. The data is automatically exported to Excel for every image analyzed. It includes a total cell count and a total positive cell count. This can significantly increase the throughput of your analysis if you're not manually having to click on every positive cell. It can also increase the statistical significance of your analysis. And then this sort of additional image, the pseudo-colored image, will allow you to confirm whether the numbers in the actual data actually match your visual assessment of the image. If you have any further questions about this type of analysis, please feel free to visit our site.